This is part 2 of 2 on chapter 3, Tools of Microbiology. In the first part of this chapter, we looked at the different types of microscopes, properties of microscopes, and then we also looked a little bit at different types of staining. This part of the chapter is going to really focus on other parts of those 6 I's of microbiology. So just to remind you, here are our 6 I's of microbiology. We're not going to go into a lot of detail on each of these. You can actually find them in your book to help you finish with your homework packet. So we kind of focused on the inspection part and information gathering a little bit and identification in the previous part. So now we're going to be looking at isolation techniques and inoculation techniques as well. So looking at how to isolate colonies and how to actually inoculate or use different types of agar to grow your bacteria or your specimen that you collected. So we'll start off with isolation techniques. So isolation, a lot of times when you get a sample, you have lots of different bacteria species mixed into that sample. So you're going to take that sample, you're going to spread it out on a plate or put it in media, for example, and then put it on a plate. Each of these different bacteria species, they will get onto the plate and then they'll divide and grow. So they'll go through binary fission, and then you'll end up with something called a colony. So on our plate we have white colonies and yellow colonies. And you can tell colonies apart. They're going to have different colors, different sizes, different shapes, different textures. And we'll look at colony, or the macroscopic part of colonies in one of our labs. So because they look different, these two colonies look different from each other, we can assume that they're different species of bacteria. So some isolation techniques, you can use something called a streak plate technique or a 4-quadrant technique, and we're going to be using this in lab, so you get to practice this. But the streak plate technique, you take a sample, you put it in one corner of your plate, and then from that one corner you spread it out into the 2nd quadrant, the 3rd quadrant, and the 4th quadrant. And you're not adding any more bacteria to your plate, you're just spreading it out or diluting down your sample as you go around from your 1st quadrant to your 4th quadrant. Another type is called a pour plate technique. So this is where you put your sample into a liquid or a broth, and then you're going to dilute that broth down and put those samples on your plate. So you have a sample that has lots of colonies, lots of bacteria. If you dilute it down, you end up with fewer colonies on your plate. And then hopefully you can see the 2 different types of bacteria in your sample. A third type is called the spread plate technique. This is where you take a very tiny amount of your sample and you spread it around using a hockey stick. So you spread it around on the plate, and then hopefully it's dilute enough where you get 2 different colonies, you can tell them apart. Each of these 3 isolation techniques, if you look on the far right-hand side, you can see that each of these techniques you end up with individual red colonies and individual yellow colonies. And then from that you can pick up one of those colonies, and that's called the isolation. You can grow that specific bacteria species from then on. So after isolation, you can also do inspection. So if you have a single species growing on your plate or on your slant or in your agar, that's called a pure culture. So all of the colonies look the same. But if there's multiple species, then you can have a mixed culture. And mixed cultures, usually we want to isolate out the different species of bacteria. Other times you can have something called contaminants. This is something that we don't want at all. And it's usually some type of mold, or it can spread out, or just appears in one place on your plate, not everywhere on the plate. So if we look at the images down here, so if we start on the left, so image A on the left, we have yellow colonies and kind of a whitish cream colony as well. So this is a mixed culture for image A. Image B, we have orange colonies, but then we have kind of this whitish blob. That whitish blob is called a contaminant. So your culture is mostly pure, but it is contaminated. And then image C, these are agar slants. They're not just flat plates. They're slants in a tube. And we have 3 tubes that each have different type of bacteria. You can tell by different color, different growth pattern. These are all pure cultures. I don't see any mixed cultures or contaminants in these. Identification of a microbe. So once you have your pure culture, you've isolated it, you want to identify what that bacteria is. So we usually use this when you want to identify bacteria species that causing an animal or human to become sick. So we can identify microorganisms based on their cell and colony morphology. So you can look at the colony, write down what it looks like, if it has any color to it. You can take a sample of your colony, you can stain it, so you can use simple staining, you can use gram staining, so some of those differential stains we talked about. Another way to identify a microbe is to sequence the DNA. You can also do biochemical testing, so looking at how that species of bacteria reacts to different environments or to different chemicals that we're going to be testing. And biochemical testing, we're going to do a big project the second half of the semester. You can also do immunological testing, and we're not going to do that in lab, so I'm not going to talk about that, and you can read about it in your book. Finally, I want to talk about media. So in order to grow bacteria in the lab, you need to provide those bacteria with nutrients. So media can be classified according to 3 properties. So it's classified based on the physical state, so you can have a liquid, which is called a broth. You can have semi-solid. This is what we're going to be working with the most, so agar. It's kind of a gelatin-like substance. You can also have solid media, which we're not going to be using in lab. Most of ours are semi-solid. We'll also be using a few broth or liquid medias. You can classify media based on chemical composition, so we have synthetic media, it's chemically defined, or we can have complex media. And then last, you can classify your media based on what it actually does, what the function of it is. So we have general purpose media. This is nutrient agar or tryptic soy agar. It grows pretty much anything. We can have enriched agar, where we add something to it, selective agar, differential, anaerobic agar or media, where we grow it in areas with no oxygen. Transport media, if you're taking a sample and sending it to a lab. Assay media, enumeration media. So we have lots of different media that can be used for lots of different functions, depending on what you need to do. So we're using mostly semi-solid media in the form of agar. So this is a commonly used solidifying agent. It's kind of like gelatin. So agar is solid at room temperature, which is convenient. It liquefies at boiling and above, so you can heat it up, you can liquefy it, you can pour it into a plate, you can pour it into a tube and cause it to solidify at a slant. And it re-solidifies at about 42 degrees Celsius. So as it cools, it solidifies. So the agar, it does not provide any nutrients. It just provides a framework where we can hold moisture and hold nutrients. So the agar itself is not actually used by the microorganism. You have to add some type of food or nutrients to the agar. So commonly used media, I mentioned nutrient agar. We also have nutrient broth. So nutrient broth is just a liquid media. It contains beef extract and peptone, so it has protein in it. The cells use that protein to grow. Nutrient agar, it's solid media or semi-solid media. So it has beef extract, it has the peptone, and then it has agar as well. For the chemical composition of the media, you can have synthetic media. So this contains pure organic and inorganic compounds in an exact chemical formula. So synthetic media, we know exactly what is in this media. You can also have complex or non-synthetic media, where it contains at least one ingredient that's not chemically definable. So you can have synthetic or non-synthetic media. We have general purpose media. This is like the nutrient broth or nutrient agar I just looked at. This grows a broad range of microbes, and it's usually non-synthetic, and it grows pretty much anything. Another type of media is enriched. So these contain complex organic substances such as blood serum, hemoglobin, or special growth factors that are required by fastidious or picky microorganisms. So enriched media, you have to add something more to it to get certain microbes to actually grow. So examples of enriched media, we can have something called blood agar, which we're going to be using in lab. So blood agar has blood in it, it's sheep blood, in with the agar, and some bacteria are going to break down the blood cells, and that's called hemolysis. So if a bacteria species can break down blood cells, it usually can cause some type of disease. So we need to watch out for these microorganisms that can cause hemolysis or destruction of red blood cells. Other species of bacteria do not destroy red blood cells, and that's shown in the image on the right-hand side. So these bacteria cells, they produce colonies, they can grow, but they don't have a clearing around the colonies that shows hemolysis. Some media are selective and other types of media are differential. So selective media, they grow one type of bacteria and not another type. So it's selective on what it can actually grow on this agar plate. Differential media, this causes different species to grow, but they have different reactions to the media, so usually they turn different colors. So selective media, we use this when we want to grow one type of bacteria, we want to inhibit other types. Differential media is when we want to be able to identify different bacteria species on the same media. Some media can be both selective and differential at the same time. The one that we're going to be using in lab is called McConkey agar. McConkey agar is selective because it grows gram-negative bacteria, but it does not grow gram-positive. So it grows one type of bacteria and inhibits another type. It's also differential. So those gram-negative bacteria, they can either grow white or pink. So if they grow to be white, that means that that species cannot ferment lactose, it can't break down lactose. If it grows to be pink, it means that gram-negative bacteria can ferment lactose, so it's lactose positive. Other types of media, you can have reducing media. So reducing media, it contains a substance that absorbs oxygen or slows down the movement of oxygen. So these reducing medium, specifically a thio media, THIO media, it's used for growing anaerobic bacteria. So bacteria that do not want to be in oxygen or they need a reduced amount of oxygen. And then another type of media is called carbohydrate fermentation media. So these different types of media, they have sugars that can be fermented. Once it's fermented, it's converted into an acid, and that changes the pH. And then your tube can actually change color. So I'm not going to talk about these a lot. You can, again, read about them in your book to learn more about them. We're going to use these in the second half of lab when we get to our unknown project.